presentation, and I do apologize for it. Cover crops is a topic that we haven't covered and continuing at in quite some time, and I thought it was about time to get these concepts out and dust them off. And it turns out there's a lot of newer and very exciting stuff that's going on with cover crops. So this is going to be a very broad discussion of cover crops in many, though not necessarily all of their uses, because trust me, some people are getting very creative in terms of how they're using cover crops to deal with the various issues they're being posed with on their operations. All right, so first of all, I want to say that more or less by agreement, cover crops are defined as a crop that's grown but not for harvestable product. And cover crops, in essence, as a term and an idea, evolved from a concept many of us knew from our training years ago as a green manure. And in many people's minds, the difference between the two is that green manures are incorporated into the soil and cover crops are not. But as some of you know, that's not necessarily true either. There are people who deliberately incorporate cover crops. So it's a very, very mushy distinction between the two. But certainly if you look at the number of publications that talk about green manures 50 years ago, 40 years ago, and cover crops and today, the term green manure appears to be dropping off people's radar screen, and the term cover crop has replaced it. And I do want to say that this knowledge of crops being grown not for their harvestable product, but for some other reason, is one that humans have known for literally thousands of years. I just mentioned Virgil here because I had to read him when I took Latin 4. He wrote what is essentially the encyclopedia of agriculture, animal production, crop production, soil information. It's a very, very long thing. They knew then that crops that they described as alfalfa and clover and lupine increased the wheat yields, that they could not just grow wheat year after year after year. They had to include specific crops such as these in the rotation in order to maintain their yield. And of course, anybody that knows the Greek literature knows that all the Romans did was steal their ideas from the Greeks anyway. And in fact, there are Greek authors that talked about this 200 BC. So we've known as a species for thousands of years that there is a need to grow crops for not through their harvestable product, but for helping subsequent crops. In Maryland, and I know some of you know that I love agricultural history, Maryland has a long history of agriculture, and it actually has a fairly substantial history with cover crops too. They're a hot tamale right now with water quality issues, but I just wanted to say that because Maryland was one of the early colonies that was settled, and because the way the colonists paid off, in essence, the loans for coming over here was by tobacco production. And tobacco was grown as long as it could be before yields declined, and that tended to be a period of 2 to 4 years. It was done with very clean tillage, and it was done, of course, in the part of the state where colonization occurred first, and there were very severe erosion problems. And it was only after people grew tobacco for 2 or 3 or 4 years at the most that they switched to other crops like wheat or corn. Now, the record shows that when that switch was made, the wheat yields were 10 bushels per acre, nothing that we would consider even bothering with today. And I do want to point out that one of the sources of information on what has happened in our region years ago is this wonderful book written by Dr. Avery Craven in 1965, Soil Exhaustion as a Factor in the Agricultural History of Virginia and Maryland. And he basically went from colonial days up to 1860. Now, this gentleman is a professor of history, and I have no idea why he got off on an ag history in our region, but he did. And his book is an invaluable source of information. And you might wonder, how do we know what was going on then about these issues? Well, people wrote letters then and communicated back and forth. And starting about 1800, there were also a lot of agricultural publications. You know, there was a magazine, I'll call it, called American Farmer. It also turns out that people did a lot of traveling. Not only people in the Americas, but people from Europe would come over, and they would write travelogues or articles. So this poor gentleman just dug into all kinds of resources to tell the story that he did. Right? It turns out that, let me see if I, yes, by the time of the Revolution, we already had serious soil depletion both in Maryland and in Virginia. And there are actually letters written between Jefferson and Washington that bemoan that problem because of the problem on the local populations. People who could move west and claim better land did, and they left behind the people with fewer resources. Jefferson and Washington, in their letters to one another, mentioned that farmers needed to grow grasses and legumes in their rotations with the cash crops of tobacco, wheat, and corn. They also thought that it would help the food sources for the human populations by being able to feed animals better. It turns out that animal production during colonial and revolutionary times was basically put the animal out in the woods or the swamp, and there were tremendous losses of animals over the winter because they literally starved. No one bothered to grow forage for them. So Washington and Jefferson saw rotations as an idea to not only increase crop production, but also to feed the human population better. There were actually towns that were and areas that were just being depopulated because the land could not support people. And although they started writing and talking about this in the 1790s and ag publications started in the early 1800s, Dr. Craven also observed that it was the 1860s before crop rotation was actually widely practiced in either Virginia or in Maryland. When Dr. Weissmuller retired, he went through a lot of his publications, and he actually gave me two treasures that I didn't even know existed. The pub on the left was done in 1957, and it's a very thorough study of the use of cover crops on tobacco production. And they look not only on yields, but they look at all the marketable aspects and actually do an economic analysis of which cover crop did the best job in terms of producing tobacco and making money for the farmer. And they were extremely thorough in their work. It's a very delightful pub. We're going to scan that and put it on our publication list because a lot of these old things have been lost, and I really hate to see that. The pub on the right is one from 1942. Now, as you all know, we were in the midst of World War II in 1942, and any nitrogen around was going to be used for the war effort to make bombs and ammunition and dynamite. And so folks in the agronomy and horticulture department, I guess it was one department then, wrote an extension pub. It's just a little 4-page thing that's warning farmers that they need to either grow leguminous cover crops or get some manure to supply the nitrogen to their crop this year. So those, I found those to be of great interest. So enough for the history. I'm going to talk about cover crops from the point of view of 6 of the functions that people use them for. One, of course, is erosion control, and I believe that's why they're called cover crops, because they cover and protect the soil surface. Another is as a source of organic matter, that gold material that soil needs in order to function properly. Third is as a nitrogen source, and I'm going to focus primarily on the legume rhizobia symbiotic stuff. Jude told you about some of the other ways that nitrogen is brought into an agricultural or an aquatic system. Scavenging nitrogen, and that certainly is the emphasis today in the cover crop program, where we are growing crops to take up the nitrate that's in the soil profile in the fall at the end of the summer growing season. And I'm also going to talk a little bit about using it for pest control. There are folks that have looked at it from a disease and also a nematode control point of view, and also from compaction reduction. That is an effort that's been led really by scientists here in Maryland. So first let's look at erosion control. I've got a little slide down there of a wonderful, vigorous cover crop. The beauty of it is if you get it in on time and you're lucky enough that year to get adequate rainfall, that you really will get a very protective vegetative cover over the surface. What you want is a good stand, and you want rapid growth so that your soil is protected during the winter when we'll still get a substantial amount of rain, and we want that soil surface protected. Now, from my look at the literature, it appears that everybody knows that cover crops are so good for erosion control that people talk about it, but they rarely actually measure the difference. So I was able to find a study from Missouri that was done in the late 1980s where they looked at some of the things they were looking at as cover crops, no cover, where they actually deliberately kept things free of any weeds, chickweed, which was their prominent weed, and then a brome grass, which is what they were using for cover crops then. Their concern was protecting the soil surface over the winter after soybean crops. Soybean residue is less in quantity than corn grain would be, and it's also a very fragile residue. It doesn't typically make it through the winter, and they were concerned about controlling erosion in soybean fields. And you can see here that with no cover at all, actually getting rid of the weeds, they had about a loss of a little over a ton per acre per year. If they let the weeds flourish, it dropped down considerably, and if they grew brome grass, they were down to about 1/10 of what it was when they left the surface unprotected. So I think cover crops for erosion control are a widely acknowledged, widely understood role for cover crops. Now, cover crops for organic matter is why many people use them. Of course, we get inputs, carbon compounds from the roots, exuded from the roots, given off by the roots while the crop is growing, and then we get it from the above ground part or the shoots when the cover crop is terminated. And there's a lot of information out there about what cover crops do in terms of their benefits, and people will say, well, it improves the physical and the chemical and the biological properties, but essentially it does all these things because it puts carbon, food for microbes, into the system, right? Of course, the plant roots themselves and the fungi actually can physically wrap themselves around some particles and hold them together. But the real critical thing is soil microbes live in a state of near starvation, and when you add a organic carbon material, those microbes are going to use that as a food source. And we start this wonderful cascade of effects that flow from feeding your microbes. Both plant roots and microbes exude materials that various scientists have called binding agents or cementing agents, things that hold particles together. Right? Many people call them mucilage. Now, those of you that are out there that are middle-aged and above, remember that golden-colored stuff we used in a little glass bottle with a rubber tip on top called mucilage, and you remember how sticky that was. Well, that's what people are implying and trying to communicate to us when they call these materials mucilages or cementing agents. And what happens is soil particles are held together into large aggregates. Now, that's good because it improves soil structure. It gives you adequate macropore space. It makes your soil more friable. It makes it easier for the roots to push through it. Of course, it decreases the bulk density and increases the pore space. It increases your infiltration rate, so what rain you get actually can move into your soil and not run off. It increases the rate at which water moves through the soil, the hydraulic conductivity, and ultimately we sort of describe all this as improving the soil tilth or improving soil quality. But it does it because of the ultimate effects of organic carbon compounds on the stimulation of biological activity in the system and all the chemical and physical effects that flow from that. Okay, here's a slide with two photos on. One is the picture of some rye, early growth. This is probably in the late fall, and you can see how those roots themselves wrap around. First of all, there's a lot of them, and they wrap around soil particles. If you've ever pulled up a bunch of grass or a bunch of rye, you know that you've got to shake the living daylights out of it to dislodge the soil that's on it. Well, we may not have the same appreciation of what soil fungi do. Now, on the right here is a picture of a soil fungi that was actually allowed to grow out on a flat plate so they could take a picture of it. But you can see all those little like fingers or hands. If they were in a soil system or a porous system, they would literally wrap around things. And if you've kicked open a log or something that's decomposing or messed around in the litter on a forest floor, you know what I mean by that fungal mycelium and meshing things and holding them together. So one of the first things that happens, of course, when a crop is growing or when a carbon source is put in is that both the plants and the fungi start growing and wrapping around many soil particles. All right. Now I want to talk for just a little bit about what happens when we add fresh, relatively succulent residue to a soil, because it's a whole series of things, and I'd like us to appreciate the many facets of it. So let's first look at, I'll get my pointer. Let's first look at the number of soil creatures and the carbon dioxide evolution. And of course, they're intimately related. The more creatures there are, the more respiration there's going to be and the more carbon dioxide there's going to be given off. So if material was added right here and the microbes were at a rather low level, give them some food to eat and they're going to reproduce to very, very substantial levels. After a while, of course, they will live out their lifespan, die, and the numbers in the soil will drop off again. But you can see that over here on the right side, we have a higher level of microbial activity, slightly higher than what we started with. And the carbon dioxide evolution, which is in direct relationship to the number of creatures, tracks the number of creatures. So we have a flush of biological activity in terms of microbes. And remember, they're the creatures that are producing some of these mucilages that hold soil particles together. Now let's go down and look at some of the other materials. First of all, there are identifiable compounds in the initial tissue, in the original tissue. Initially, of course, everything that we put in is there, but over time, those materials are used as a food source for the microbes that are in the soil, and they very dramatically decline. Now over here on the right side, we'll see that there still is some stuff there. That's probably represented by the more resistant compounds that there aren't many microbes around who can access. They don't have the enzyme systems. They can eat those things, but they sure ate the things that were easily digestible and that they had the enzyme systems for as soon as that material was put in the soil. Now, notice also that as this is going on, there's a lot of carbon dioxide being given off, okay? All right? As they consume the material in the residue in the cover crop, CO2 is given off. As they consume those materials, there's some intermediate things being produced, synthesized by the microorganisms themselves. And this graph shows how some of the new stuff becomes a different compound. It's been changed. And some of that, not all, but some of that may actually contribute to the formation of humus. Okay? Some of it, of course, is of this newly synthesized stuff is also given off as carbon dioxide. So over here at the other side, after having added a material, you have a little bit of the original stuff left, probably the more resistant materials, and you've got a significant, though not huge, increase in humus. And the reason I like this graph is because a lot of times people have a sense of how many pounds of carbon they're adding in their cover crop, or their manure for that matter, and they seem to think they should get a commensurate increase in humus. But the fact is, only a portion of the carbon you start with in your organic addition, regardless of what it is going to end up as humus. A huge portion of it, especially if it's a fresh material, is going to be given off as carbon dioxide. And Craig's question is, how would the graph be different if it's killed and left on the surface? Well, this is going to happen at the surface. Material doesn't have to be buried for this to happen. As it interfaces or hits the surface, some of this is going to go on in that in that layer of soil right at the surface. Okay, so Adding carbon to our system is good, and a lot of things happen when we add it. Now I'd like to talk for a minute about cover crops for nitrogen acquisition, and this is nitrogen fixation, as Jude was speaking about. And I'm only going to focus on the legume rhizobia teams, those symbiotic relationships between the two, because if you look at the pounds of nitrogen that's fixed by the different kinds of nitrogen fixation he talked about, it's the symbiotic ones between legumes and rhizobia that are capable of fixing the most nitrogen per acre. Now, people are talking about the nitrogen fertilizer equivalents of manure, of cover crops. That's pretty close to what we would call a nitrogen credit. And one thing I want to mention is sometimes in books and periodicals you'll see tables of how much nitrogen is fixed by the legume, but don't kid yourself into thinking that all of that nitrogen that the crop fixes in its above ground portion is going to be available, because remember, it also fixed carbon, and as the microbes use the carbon, the energy stored in the carbon-carbon bonds, they're going to use up some of the nitrogen in the cover crop in their own metabolism and in their own cell building. So only a portion of what's actually fixed will be available to the subsequent crop. And that's what people now refer to as the nitrogen fertilizer equivalents and what we commonly know of as the nitrogen credit. Now, the organic producers have found that they even look at something that's drilled down to a little bit lower level than that, and that is nitrogen delivery. If you're in a system where you're only mechanically killing your cover crop and you may have issues with weed production, the fact that your legume may have fixed the nitrogen doesn't ever mean that it'll get to your next crop, right? You've got to make sure that you have adequate weed control and that you've killed your legume and it's not reseeding and using up moisture. So there's a very interesting paper out of North NC State and some people working on organic production that now use the term end delivery. And that essentially refers to the nitrogen you can count on from your legume given the tillage and weed control system that you're using. Okay, I'm going to talk most about hairy vetch because, quite frankly, it is the winter legume of choice in the mid-Atlantic. Most of the work in terms of fixing nitrogen in rotation, both in Pennsylvania, Maryland, and Virginia, has been done on this crop. And it turns out that it has its maximum nitrogen contribution at flowering. That's when it's got the highest biomass, and it's when its nitrogen content has reached its highest point. Now, flowering is also the best time to terminate it if you're using mechanical termination. So again, if we go back to sustainable organic systems, they are not using herbicides. They need to mechanically terminate it. And it just turns out that using the same implement, you get much per die-off when it's growing vegetatively than you do when it's flowering. So people would like to get the most nitrogen out of the money they've invested in buying the seed, planting the seed, taking care of it, needing to kill it. But it turns out that it's been difficult to do that because if you let vetch go, most vetch varieties go until they've gotten 50% flowering, you've lost a couple weeks of your corn season. You know, it interferes with getting your corn crop planted on time. So I do want to point out that some work at USDA by the same group that Jude Mall is in now was done by his predecessor, Dr. Teasdale. He looked at some cultivars of hairy vetch that a plant breeder at Auburn developed. Not genetically engineered, just traditional plant breeding. They call these the Auburn cultivars, and they found that they are winter hardy here in Maryland, though not necessarily in Pennsylvania and not in New York State, and that they flower 2 weeks earlier. So, you know, one of the things Jude started to talk about at the end is that we haven't spent the time breeding cover crops the way we've spent time and effort breeding our main crops. Well, some people are working on that. At Auburn. The Auburn cultivars have been tested here, and getting that 2-week jump on being able to get your vetch reaching maximum nitrogen contribution point and still be able to get your corn planted could be a real plus. So curious as I am, I tried to get my hands on some of this last fall, and none of the ag suppliers that live in an area where I would frequent know anything about this. So I guess if we're going to use these, we need to start early and find a source of seed. But for those who are interested in using Vetch as a nitrogen source for corn, particularly in organic systems where your legume source is the primary source of your nitrogen, they probably want to get a hold of some of these improved cultivars. Now, one of the things that's always frustrated me tremendously, and I know it has frustrated some of you, is that for our winter perennial or our winter annual legumes, we have only very wide ranges of nitrogen values for the nitrogen credits. And people have often said to me, well, what should I use? Harry Vetch says 75 to 150. Should I, when is it 75? When is it 150? Well, the problem is that we were given only very vague data. Well, it depends upon when you planted it and how much growth you've got and when you kill it. Those are pretty vague, hard to operationally define. Well, again, Dr. Teasdale out at USDA looked at, first of all, does Vetch respond to, in terms of flowering, does it respond to day length or does it respond to temperature? And it turns out it responds to temperature. And he found a very good relationship between growing degree units, which happens to be a preferred term for growing degree days, at a base temperature of 40 degrees Fahrenheit to predict when Vetch would reach its peak in terms of nitrogen supplying. He found that it takes about 926 growing degree units after you plant it to supply between 120 and 140 pounds of nitrogen, and that's probably what many growers need for what they're going to plant next. Okay, so my only concern here is that there doesn't appear to be a service, an ag service, a website or anything that's tracking growing degree units at these various base temperatures that we use for pest control or wheat fertilization or in this case, Vetch termination. So this certainly represents something we should encourage be done in the near future. Okay, one thing people often ask is, how do we know what the legume credit is? How do we figure that out? And I want to tell you that we don't do it by a soil test. We do it by having replicated plots on a uniform area where we on some grow the cover crop, where we on others don't grow the cover crop, and then after both of those conditions, we impose a nitrogen rate experiment on both areas. Okay, and this slide shows some work that has been done on a summer legume called sun hemp, and this is in Alabama, but it's a nice graph, and that's why I'm using it. So you can see this the black line represents the response of corn to the various nitrogen rates, and this is 0, 50, 100, and 150 in English units. You can see that when no nitrogen was applied, we got a very low corn rate, when 50, 100, 150. So this is the response curve for the land that had no cover crop at the various rates we were looking at. Then if you look at the response curve for what the corn yield was at those same rates, 0, 50, 100, and 150 pounds of nitrogen on the area that had gotten sun hemp, you see that zero nitrogen sun hemp produces 6 megagrams per hectare when zero nitrogen on the no cover crop was 2. Okay, well what you do here is, let me see if this is going to work. Yes, you go over to, you start where the yield at 0 nitrogen is on your legume cover crop. You go over to where it meets the response curve for no cover crop, and then you drop down to your horizontal axis, and that amount represents the nitrogen credit. So in this case, they got a credit of about 100 pounds of nitrogen per acre from the sun hemp crop they grew. Right? Now, in order to feel comfortable with giving that out as information, this would have to be done on multiple sites. and it has to be done multiple years. So I hope you can appreciate the fact that this is not quickly or easily done. In Maryland, we'd probably want it done at Wimwreck, at Clarksville, at Y, at Salisbury, and we'd want it done for at least 3 years, and then we'd see what the variability is, and we'd come up with some ballpark figure. Or, in the case of what we have on our info cards, a ballpark range. Right? So it's not a rapid process. Okay, now I want to talk a little bit about cover crops as nitrogen scavengers. This is certainly why cover crops are encouraged in Maryland to use up that nitrogen that's left at the end of the summer growing seasons and get it out of the soil so that it doesn't move into the groundwater during what I'm going to call the leaching season. And the best crops to use here are the non-legumes. You know, you want something that is not going to have any possibility of taking care of its nitrogen nutritional needs itself. You want it to take up everything it's going to get from the soil. And the fact is, when the nitrate is incorporated into the cover crop biomass, it's protected from leaching. It's in the tissue, and that's where we want it. Okay. Now, why do we have a problem here? All right. Well, a problem. We have a challenge here. Okay. First of all, this is a pretty good representation of the soil hydrologic cycle. This is from an Eastern Shore location. I believe Dr. Meisinger got this information from work he had done down in Salisbury. All right, and so if we look at precipitation, that's the blue, we can see that unlike some parts of the world, we don't have a wet season and a dry season. We're not in Southern California. We're not in Italy or Greece. We get approximately the same amount of rainfall all year long. No rainy season. All right. We can also see, looking at potential evapotranspiration, that there isn't much in the winter. You know, the sun isn't very strong, so there's not much evaporation. Our crops, for the most part, are not actively living or they're dormant, so evapotranspiration is very little. But as it starts to warm up and plants start to grow, come out of dormancy, are planted, whatever, the potential evapotranspiration reaches its peak in the summer and then drops back off again after things go dormant or they die or they're harvested. All right? So also realize that because we've got rainfall coming at a more or less constant rate all year long, our soil profile tends to get filled up in the late fall, and during the winter it's full. All right? It's at field capacity. So if we get more rainfall, and of course we do, water's going to move down into the soil profile. So this, in essence, is when our soil is full of water. If we get more, it's going to leach down, move down. And this is January, February, March, maybe into April. And then in the summertime, when our crops start growing and things come out of dormancy, our soil isn't full of water anymore. There is space for that water to go when it rains. Right? Then we get to the fall, our crops are dormant, harvested, whatever. It rains, it starts to fill up the soil profile such that in a typical year by mid-December, our soil profile is full. Right? Now the consequences of this is that we're going to get some drainage, this red thing here. We're going to get water moving down in the soil profile during the time of the year when our soil is at field capacity. It's full. It can't hold anymore, but we're still getting rain. And anything that's dissolved in that soil water is going to move with it. So this is, in essence, our big leaching season, our winter, from typically mid-December through maybe early March. Then drainage or leaching is going to drop right off. We get only minimum leaching typically in rain-fed agricultural areas during the summer, and then it might pick up again going into the late fall. And the only case around here where I've seen much discussion of leaching in the summer is you could get some flow through macropores. All right, that in a heavy rainfall don't get an opportunity to get stored in the small pores. And some work in Delaware clearly shows that in irrigated situations, if you get a substantial rainfall and within a few days of having irrigated to field capacity, that you will then get leaching. But in a rain-fed situation, we're pretty much here in the summer. Okay, so if you want to optimize scavenging, what do you grow? Well, you certainly grow a non-legume. And certainly during the 90s, there were some big discussions as to what was better, Is rye better? Is wheat better? Is barley better? And there certainly was a belief for a while that rye was really the only thing to use. So there was a flurry of work looking at some at other small grains other than rye that perhaps could be grown for their grain in addition to being scavengers in the fall. Small grains like wheat and barley, which are a little easier to manage. Les Vaux has been quoted as saying that in 5 days, rye can go from boot to heading. And if boot is where you want to kill it and it happens to rain when you should be out there killing it, it can be out of control by the time you can actually get out in the field again. So there are people who had some really bad experiences with rye and who were reticent of using it, even though folks were saying, well, it's a good nitrogen scavenger. So I'll show you a little bit of data where people have looked at some of the other small grain species. I'm going to show you some data on the effective planting date. It's very clear that the earlier you get it planted, the more uptake there's going to be. That's been shown time and time again to different extents in different places, but it's certainly a fairly well-established fact. And then a little bit about kill dates. Certainly your carbon nitrogen changes. We now have some guidance that shows that we might be able to use growing degree units in addition to going out and looking and seeing, well, is it blooming? Is it at 50% bloom? Is it still vegetative? We might have some better data now to decide when to terminate a crop. Okay, so here's an experiment that Frank Cole and some other folks did, and they looked at some various rates of manure. The rate 1 was about 150 pounds. Rate 2 is twice that. Rate 3 is 3 times that. They put the manure down for 3 years in a row prior to growing this cover crop at these rates. And you can see here that they were looking at barley, rye, triticale, and wheat. And triticale is a cross between rye and wheat with the help of a chemical called colchicine that doubles chromosomes. So you can see that in Queenstown in 1997, these NSs, this all means that there was no significant difference between these 4 species in their nitrogen accumulation early in the season. All right. There were some differences at Quantico in 1998. The A means it's different than the B's. But again, rye doesn't necessarily come out as being the only thing that's effective. It's pretty clear that any of these small grains will take up nitrogen, and certainly if you have a reason to grow something other than rye, there's a justification for doing that and still considering that you have a fairly decent scavenger. In addition to that, Jose Costa, he's our wheat breeder here at University of Maryland, and Herman Delero, our former cropping systems person, and Frank Cole did a study where they looked at 25 different wheat varieties and compared it to the only cultivar of rye that was on the market at the time, and that was Wheeler. Right? And they found that there was very little difference in early season nitrogen uptake among the rye and the 25 varieties of wheat. The rye took out 34 pounds of nitrogen, and this is by mid-January. All right? A third of the wheat varieties actually were higher than the rye, all right? And the average wheat variety was not that different from what rye was, 31 pounds. So again, no huge reason for choosing rye over wheat. No reason to think that wheat is inferior, certainly. Now, they didn't let the rye go to physiological maturity. They killed it at the optimal time as a cover crop. They did let the wheat go to maturity, and they harvested it, and they found that there was about a 20 pound per acre difference in uptake between the different varieties, a difference between 60 pounds per acre of nitrogen and 80. So it's clear that there are genetic differences among wheat, and there may well be among rye too. It's just that most of the cover crop rye out there is what's called common. It isn't a variety. The only variety out there, in fact, is Wheeler, and that's the only one really that's been included in most of the studies because you know a little bit about it and how it's behaved for other people in other situations. All right. Now I want to show you some data here on planting date, and this is essentially a graph that's showing you how much nitrogen was accumulated And this is in kilograms per hectare, but that's, if you multiply this times 0.9, you get pounds per acre. So for all practical purposes, think of these as being almost the same. The red line represents the uptake when the rye was planted at the Y on October 1. The green diamonds represent October 14, and the black or blue squares represent October 30th. And you can see that at any point along the way here, the planting date made a big difference. So when people complain about, oh, those early planting dates, I don't know why they're choosing them. They're choosing them because in situation after situation, we find that there's more uptake when you plant early than when you plant later. Now, I will say that these quantities, and that's a little off the graph, these were between about 80 and 160 pounds of uptake by March. These are the highest uptake levels anybody's ever reported. You'll see some other ones today that are much more modest. I don't know what was unique about this situation that led to such massive uptake of nitrogen, but it certainly dwarfs anything we've seen on other sites. Okay, this table's a little bit busy, but there's a couple things I want to point out. First of all, we're looking at dry matter, how much dry tissue, nitrogen uptake, pounds of nitrogen that was taken up by that tissue, and also the carbon-nitrogen ratio. All right, you probably remember from many of our discussions about organic matter incorporation is that you have to keep an eye on your carbon-nitrogen ratio. And as a general rule, and this is a very general rule, once you get the carbon-nitrogen ratio above about 25, you've got to watch out for immobilization. Because as the microbes use the carbon in that material, they're going to tie up nitrogen beyond what's in the tissue and take it out of what's in the soil. All right? So if we look at early winter kill, late March kill, early April, and late April, we can see that the dry matter kind of chugs along here in the winter, shoots up once it starts to get warm, and by mid-April, it's reached its max. And this is about the time it was terminated. Right? You can see that the pounds of nitrogen, of course, track that. You can get about 43 pounds of nitrogen taken up here by late April as opposed to the early winter of 20. And this, again, this is what I'm pointing out. These are much more modest amounts of nitrogen uptake than that experiment that was done at the Y in the late 80s, early 90s. Now, look at here. Look, Piedmont in 1990, carbon-nitrogen ratio 15, that's nice, 20, 21. Until we get to the late April kill date, we have a carbon-nitrogen ratio we're probably pretty comfortable with. At this point, we're probably concerned. Okay, not that we know it, but we should be concerned because we're well into the immobilization range right now. Are there differences between the coastal plain and the Piedmont the same year? You better believe it. Okay, you can see that somewhat more dry matter and nitrogen, somewhat more dry matter, less nitrogen, believe it or not. The Piedmont stuff was richer in nitrogen, even though it had produced less stuff. But what you really notice is the difference in weather from one side of the bay to the other, Clarksville versus the Y. And so at the same point in time, the cover crop at the rye was much more mature and had a much wider carbon-nitrogen ratio. And this is why these general statements that people try to make statewide, well, you need to kill it at this time. The time is going to vary from year to year, and the time is going to vary from location to location. And that's why if we can work out a tool like growing degree units to know when to kill these crops, we would be much, much better off. Now, I am going to show you a graph later on, or a table, where we look at the coastal plain conditions in 1990. So remember that we're looking at both situations, early April and late April, where our carbon-nitrogen ratio was pretty wide and well into the range where we would expect immobilization. Okay, now here's a study, and I just want to make the point of how we establish our cover crop is important in terms of how much nitrogen is going to be taken up. So Bob Cradiville and his group just published an article in 2011 on work that was done for several seasons before that, and they were looking at the difference between the nitrogen uptake in wheat, or if it's in parentheses in rye, the next spring, and this is at Beltsville in 2007 and 2008. And they compared broadcast, they just broadcast the seed, no incorporation, no nothing, just broadcast it, no-till drilled, and disked. Okay, now they only disked the wheat treatments, not the rye treatments. I don't know why. Next time I see Bob, I'll ask him. So we don't have a comparison for all of these. Now the early establishment was that October 1st date, and that's that date that the Y experiment showed was so effective. And the later establishment date was somewhere between the 20th to 20th of October to the 1st of November. Okay. Now this was not the best year for cover crops in the sense that the summer had been very dry. So there wasn't much soil moisture near the surface, and then there was no rain essentially until late October. All right. So let's look at this. If you establish it about October 1st and you broadcast wheat, by the next spring you've taken up 10 pounds of nitrogen. If you've no-till drilled it, you've taken up 25 pounds of nitrogen, and if you've disked it in, you've taken up 18. So it's pretty clear that the better soil-seed contact we get, certainly the more plants that are going to successfully germinate and grow, and the more nitrogen you're going to get taken up. Now, one thing Bob did do with his grad students is go out and count how many plants made it. Okay, I'm not showing you that data, but if you'd like to read this article, I could certainly give you the full reference. In terms of the rye at these same sites established at the same time, you can see that by the next spring the rye had taken up about twice as much. If it was broadcast, about 50% more if it was drilled, and there was no treatment where the rye was drilled. Later establishment, well, that's not as good. Okay, you can see that there was much less uptake by the wheat if it was broadcast compared to early establishment, and the same thing was true for the no-till drilled. The same thing was true for the disked. Okay, so establishing things early is better, and getting better soil seed contact is better. Okay, there's some nuggets in this table, so bear with me while I walk you through it. One of the questions that often comes up is among farmers and nutrient management planners is, should we change our nitrogen rate when there's been a wheat cover crop? And many people have looked at this, and overall the decision is no. Okay, if you're managing your cover crop optimally, you shouldn't have to do it. Now I will say that there's a little star here, and for those of you that are knowledgeable of statistics, that's telling you that this comparison here, 80 pounds of nitrogen in early April comparing a check and a rye cover crop, is the only significant comparison in this table. The LSD was 35 bushels. Okay, that's a big LSD. It's a real LSD. All right, but this data here is very informative to me, this zero data. This clearly is a situation where there was a typical corn planting rate in a location that was nitrogen poor. And you can see where the check, where there was no rye, did better than the plots where there was rye, almost twice as better. You can see that in late April, the check where there was no rye did better than where there was rye. So it's clear that there is some nitrogen from the system that's being tied up as the rye cover crop is being decomposed. On the other hand, this was a field where they were shooting for 150 yield goal, and you can see that if you're fertilizing as you should, there really is no need to adjust your rate. In the case of the early April kill, they actually was no meaningful difference between the yield you got when there was no cover crop compared to when there was. And even in the late April kill, that difference is not significant. So this is, again, kind of adds credence to other people's statements and work that says, no, we don't need to adjust our corn fertilization rate for a rye cover crop. Now, nor should we reduce it, the environmentalists tend to think that, well, if you tie that nitrogen up in that cover crop, and it's gonna be released the next year, and the corn's going to use it. Well, it's not that simple. Remember that carbon was fixed too, in addition to the nitrogen. Remember that there's some structural carbohydrates that are going to take a while to break down. And the work that's been done to try to show whether we should modify our rates down, there has been some of that done, but it's usually been in periods of time when there's been some drought and the data is very noisy. So there really is no justification for modifying the rate up or down for corn based on your cover crop. Okay, now, using cover crops for pest control. This is an extremely exciting area. I wish I could say it was more successful, but it certainly interested me, and I spent a lot of time over the Christmas break reading about these critters. There's a family of plants called the Brassicaceae. We might know those as the crucifers. They include things like canola and the mustards and rape and lots of other things we eat as vegetables, broccoli, kale, Brussels sprouts. People have looked at them in terms of being biofumigant crops. Now, remember that some of the materials that we use to fumigate soils are now off the market, all right? And when that looked like it was a sure thing, scientists started looking at alternatives, all right? The fact of the matter is that the Brassicaceae produce compounds called glucosinolates. Okay? That's a whole family of compounds. These are secondary metabolites, and that sort of means that they have a minor role in life. I'm sort of paraphrasing here, but they're not needed for survival per se. All right? These compounds actually react with water. They hydrolyze with the help of an enzyme, of course, and they produce some really nasty things. This is just one. I'll show you some other names later, like isothiocyanate. Now, I think we all know that anything with anything in isn't good for us, and it probably isn't good for anything else that's alive either. And so folks looked at these crops as potential cover crops, particularly to control root knot nematodes. Most of the work has been done with root knot nematodes. Now, there are other families of nematodes, and I'm no nematologist, but a lot of the work has been done with them. And the hope was that they could find a way of growing and managing these crops to substitute for the banned chemicals like methyl bromide. So I'm going to tell you the Brassicaceae story here over the next slides. Okay, now here's an example of the molecule we're talking about. This is a glucosinolate model molecule. This is the glucose, okay? This thing down here is a carbon-bearing amino acid, and R stands for the fact that this can be up to 100 different things, all right? We also have a little sulfate group over here, okay? So essentially, there's an enzyme called myrosinate which can cleave this right here, releasing the glucose, freeing up the sulfate, and leaving this left. Now, this isn't stable, and depending upon the environment it finds itself in terms of pH and metals, it can become one of a whole host of things. Okay, so breakdown products, isothiocyanate, thiocyanate, organic cyanides, all toxic. Different toxicities to different creatures, all right? We're not saying they're equally toxic to everything. They're not. They appear to be rather short-lived in their effect in the soil. They volatilize out. They absorb onto organic matter. And they have a bitter taste. And the plant ecologists think that these compounds evolved in this family to protect them against predation, herbivores, a creature eating them, because the materials, in addition to being toxic, also taste bad. They're bitter. Okay, so there's this enzyme called myrosinate, and it breaks it down, and it's in the cell, but it's on the outside of a vacuole where the glucosinolate exists. So the toxins aren't going to be produced until the cell wall is ruptured, and when that happens, the vacuole usually bursts or is ruptured too. The enzyme comes in contact with the glucosinolate, or the glucosinolate, and in fact the reaction happens. So in order for this these crops to work as effective pest control agents, you've got to really go out of your way to break those cell walls up to the greatest extent you can. And the folks who've tried looking at it have looked at it with a flail mower, right? That apparently chops it up into about as many pieces as you can get with available equipment. And remember also that the daughter compounds, the breakdown products, are volatile. So what guidance exists on the use of these for nematode control says, don't chop any more than you can turn under in 2 hours. And we're talking about the best incorporation you can get. We're not talking about little mix it in on the surface. You want this stuff down in the soil. Okay, so what I want to tell you is, in really controlled situations, you know, like a growth chamber or like a petri dish or like leaching columns, there's data out there that says, yep, this stuff will kill nematodes, this stuff will suppress disease, this stuff will keep weeds from germinating. The problem is when you go out into the field where you have less control, scientists have often found that they can't prove effectiveness under field conditions. Okay, so there are people who found that it reduced nematode populations when they actually counted how many they had per cubic centimeter, and then there are others that found it didn't. And even where it did, the effect only appeared to last one season. Of course, what they're really concerned about is, does it help the yield of the next crop by controlling the nematode invasion? And in some cases it did, and in many cases it didn't. So the conclusion is, theoretically, this should work. It sounds great. We know these compounds are viciously toxic, and it's probably a good idea to include these crops in your rotation, but don't expect them to be as effective as chemical treatments, because they're not going to be. Okay, now I want to tell you the cover crop for alleviation of compaction story, and this is really a Maryland story. Dr. Weil, our soil fertility sustainable ag person here, is really the one that's with his grad students done a tremendous amount of work on this. They're called forage radishes. They're also called daikon radishes. You can buy daikon radishes in the food store, particularly the Asian food stores. And some people now call these bio-drilling crops. In fact, they can create low resistance paths or channels in the soil, particularly in the compacted subsoils, that can then be used by subsequent crops when this crop dies. And folks have looked at them both alone and in combination with small grain rye. So there's at least one study out there where they compared daikon radish to rye and then together. It turns out that their effect is greater in a typical wet winter. You need to understand that soil strength is greater in the dry soil than it is in the same soil when it's wet. So if you have a compacted soil and you're using something to break up that compaction, it's better to do that breaking up in the wet season. Okay, and one thing that I did not realize until I was getting ready for this webinar is that they are very good nitrogen scavengers. And I mean very good. I'll show you some data in a few minutes. Okay, first of all, here's a picture Dr. Weil took last year. Typically, these winter kill by December or certainly January. I'm sure any that are in this year have winter killed. But last year we had a milder winter, and in March they were still alive and they were getting regrowth. So there's going to be some termination necessary here. They do start to decompose in the late winter and early spring when other cover crops are still growing and taking up nitrogen. And in fact, they actually start to break down. And Dr. Weil's research suggests that if you're going to use them, you plant an early spring crop so you don't lose the nitrogen they tied up. Right? They can be immense. You know, some of them are only 6 to 8 inches long, but some of them are 12 to 18 inches long and 4 to 5 inches in diameter, so they can really drill down into the soil. Now, whether or not they have help the next crop in terms of letting its roots penetrate into these newly formed channels, and whether we're going to see it in terms of yield increases depends upon whether the next season is moisture limiting. If there's plenty of moisture, you may not see a yield advantage. If it's if it's water limited, you will probably see a yield advantage. So it's not a sure thing. Okay, let me talk just a little bit about how they actually demonstrated that these roots went down. They used something called a microrhizotron camera. So there's a transparent tube that you essentially need to bury in your soil root zone and leave there. Okay. Then there's a digital camera that's in this thing here. It talks to this intelligent machine back here, which focuses and supplies light, and you can essentially take a picture of the root zone, and by noting where you are in terms of where you are in terms of your cord and your camera, you can go back to the same place again and again and again. So they charted on a biweekly basis where the forage radishes were, and then they came back when the soybeans were actively growing to see whether or not in some of those channels the soybeans were subsequently found. So it was it's a phenomenal tool and a very elegant experimental technique. Okay, so do they, oh, and by the way, they do have some pictures in there, some photos in the article, but I didn't think they would reproduce. If you're interested in knowing about that, drop me an email and I'll send you the citation so you can get a copy. In terms of nitrogen uptake, and this is a point I want to make, they looked at forage radish, oilseed radish, canola rape, and rye. And I hope you know the canola is really just a made-up trademark for a low uric acid rape that was bred by some plant breeders in Canada, I think Manitoba. It's just a cultivar, essentially. They looked at how much nitrogen was taken up by the shoots in the fall, and you can see that the radishes, they hold their own. They also looked at the roots. It's fairly easy to look at the roots of the radishes because they're pretty big. Notice they didn't look at the rye roots. Much harder to separate the roots from the soil. And then they came back and looked at the uptake in the shoots in the spring. And you can see that there's pretty significant uptake. 70 pounds of nitrogen were taken up in the fall by the forage radish. So it certainly is right up there in terms of impact, in terms of nitrogen uptake, with some of the things that are used more traditionally as cover crops. And I was very, very impressed with this spring data, which showed that a rape cover crop actually took up about a third more than the rye cover crop at the same site, same environmental conditions, same weather, et cetera. So it's some of these Brassicaceae are pretty heavy hitters in terms of nitrogen uptake also. Okay, I do want to make the point that cover crops aren't just for fall and winter. If you're considering using a cover crop for soil tilth, soil quality, to get some organic matter in there, there's some excellent species to use in the summer. The Sudan sorghum cross and Sudan grass are used by a grower near me who operates a vegetable production facility. And sun hemp is now being looked at as a good summer legume nitrogen fixer. Work farther south has shown that it can fix 100 to 120 pounds of nitrogen per acre in about an 8-week period. So there's certainly some times when people want to consider these. Okay, which cover crop to plant? Well, what do you want to do? Do you want to take up unused nitrogen? Do you want to deal with your compaction? Do you want to increase organic matter? Do you want to protect things from erosion? And when do you want to do this? Okay, there's cool season choices and there's warm season choices. All right, and then we get to the very esoteric thing of, well, I've got somebody that wants to grow cover crops and I need to do a nutrient management plan for them. What do I do? All right. Well, if they're growing the crop for the sake of scavenging the nitrogen and they're in the option where no nitrogen will be applied at all, then there is one crop code, and that is 51, regardless of what you're growing. It's called cover crop for water quality. And the only thing you'll get for this is a line requirement. There will be no N, no P, nor will there be a K recommendation either. Now I will say that if you're growing a cover crop for another purpose, erosion control, nematode, compaction, organic matter in an unlimited system, there are modest nitrogen recommendations depending upon the crop you choose of between 20 and 50 pounds. And realize that in many of the research situations where people were looking at the effectiveness for compaction alleviation or the effectiveness for nematode suppression or erosion control, they were fertilizing that cover crop. Okay? So cover crops can be used to scavenge nitrogen, but if you're not using them for that purpose, and if you think you might have an unlimited system, you may want to choose the crop code that actually has a nitrogen recommendation. Okay, I'm about done. You're probably tired. Up here is a picture of a forage radish. This is a rather small one compared to some I've seen. Here's a beautiful crop of vetch, and this is my favorite, crimson clover. And if you have any questions, I'll be glad to take them now. Let me see.